Good morning, 4431, Tuesday, Thursday lecture. We're going to start on 12.1, components of the circulatory system today. So we need to think of a few things. Let's talk about blood. Blood's composed of formed elements. So we first have some formed elements. These are just cells and cell fragments that are suspended in a liquid that we call plasma. Dissolved in the plasma is plenty of proteins, nutrients, metabolic waste, and other molecules being transported between our organ systems via the circulatory system. The cells, they consist of erythrocytes, or RBCs, red blood cells, leukocytes, and the cell fragments, which are platelets. We measure this like we did in our lab using a method called the hematocrit. The hematocrit is just defined as the percentage of blood volume that is erythrocytes. So we do this through a centrifuge. We get a capillary tube, we fill it with blood just like we did in our lab. We have a little tube like this. Put a little clay stopper there once we fill it up with blood, and we get our blood there. We spin it and we get different, we get a supernatant and we get the bottom, which is the erythrocytes or the hematocrit, and it'll look something like this. Let's just look at this as a centrifuge tube. And our first layer right here, we got another layer, and then another layer right here. This right here this is our hematocrit. And in males, equals 45% approximately. Females equals 42%. In between, right in here, this blue area, this is our leukocytes and platelets. And lastly, up here in the black area, this is our plasma, approximately 55%. This is also known as a buffy coat. So if you hear buffy coat, you'll know that they're being talked about. Leukocytes and platelets are being talked about. So if we look at this from the mass side of things, we can say that the average volume of blood in a 70 kilogram person is 5.5 liters. Equals, start over. Blood in 70 kg person. So we would multiply 45.45 times 5.5 liters equals 2.5 liters of red blood cells in a 70 kilogram person. We also, when we subtract 5.5 liters minus 2.5, this is 3 liters of plasma in a 70 kilogram person. So when we talk about plasma, it's important to talk about some of the things that we see in plasma. So let's just do this, plasma. Just straighten down a little bit. And in our plasma, we have proteins. Greater than 90% of plasma equals H2O. So once we have this great amount of water, this suggests that many of these proteins are soluble within it and dissolve so that they can be transported between organ systems. The plasma proteins consist of most of the plasma solutes by weight, and they exert an osmotic pressure that favors the absorption of extracellular fluid into capillaries. So that way our nutrients, waste, and such can be moved in and out of the capillaries and processed into the right organ systems from there. The proteins inside the plasma can be grouped as the albumins, the globulins, and the fibrinogens. So the albumins and the globulins have a whole bunch of different functions that we'll talk about throughout the book. The albumins are the most abundant. And they're synthesized by the liver. Globulins are used a lot in the immune system. Fibrinogens function in clotting. Serum, when we talk about serum plasma, this is just our plasma with fibrinogen removed. And the protein and other proteins involved in the clotting reaction. So that's the basics of our plasma, our hematocrit, the foundations of that. Next, we're going to talk about how a multipotent, uncommitted hematopoietic stem cell is differentiated into red blood cells, platelets, and the many other different types of proteins and such found, or cells found in blood.